Jessica?
where we describe where we both are shown in the folds, outlined by simple columnar, of course, and also we saw here your peg cell. Now what I mentioned here as the last layer is supposed to be tunica serosa, kaya lang isa dito sa slide natin, ano? That's why we said tunica adventitia. So please take note, this is actually supposed to be lined by tunica serosa. Yun lang po yung correction natin. Please correct, tunica serosa is the outermost layer of your fallopian tube. Now, the slide we're supposed to take up today is the cervix and the vagina. This is the slide of your cervix. The cervix divided into two parts. You have your endocervix and the exocervix. Okay? So the clue that you are from the endo to the exo is the shift in the lining epithelium. Look at that. Lining epithelium in the exocervix is already stratified squamous non-keratinized epithelium. Okay, so this is what we call the portio vaginalis. On the other hand, the area where it's still endocervix is lined by simple columnar epithelium. Now, what is the difference as to histology between exo and endo? Endocervix, you will still see plenty of your cervical glands. These are your cervical glands that you find in your lamina propria. Again, this is mucosa, of course, supposed to be lined by simple columnar. And these are the cervical glands which secrete mucus for lubrication. And if you study further your mucosa lining, you'll see that they are thrown into folds. We call the folds plicae palmitae. Plicae palmitae. Plicae for folds, palmitae palm-like. Palm-like folds. So yan, plicae palmitae. So note the number or numerous cervical glands at the level of your endocervix. Okay, and the secretion, take note, is mucus. On the other hand, if you study your exocervix, there you go, you see the stratified squamous non-keratinized. And if you look at the lamina propria, as you go further distally, you will see already the decrease or almost none, no more glands in this area. Okay, and then po ang difference niya, no? So this is again lining epithelium and this is lamina propria. Alright? So let's move on to your vagina. So those are the only things you'd like you to take note of. No? The shift from simple columnar, there you go, to your stratified squamous non-keratinized. Now with regards to the vagina, of course, this is already the continuation of your exocervix. And therefore, you will see your lining epithelium of stratified squamous non-keratinized epithelium. Again, in the lining epithelium, in the lamina propria, you don't see no glands here. No? So that is also a picture that is a continuation of your exocervix. Okay? Now, notice you can see muscles here. These are, of course, smooth muscles. Layer of smooth muscles. And the outermost layer, on the other hand, is not anymore serosa, but adventitia. Remember, it is already outside the peritoneum. So, outermost layer, adventitia. You see your layer of smooth muscles. You see your layer of stratified squamous non-keratinized epithelium, underneath which is your lamina propria, without glands. Okay? So this is just the continuation of the female reproductive system that you're supposed to take up last week. So today's topic is male reproductive. So the first slide we're going to take up, again, is your testis, slide number 66. All of these structures, of course, as we all know, are your seminiferous tubules lined by seminiferous epithelium. Now, when you say seminiferous epithelium, it's made up of your developing stages of your sperm cell. Okay? So again, just as a review, this is a seminiferous tubule, and again, do not forget that the highly eosinophilic cells in between your seminiferous tubules are your Leydig cells. Leydig cells, of course, are the portion that is endocrine, which secretes your testosterone. The exocrine portion, on the other hand, of course, is your seminiferous tubule. Now, as a review, what are the things that we're supposed to see in the seminiferous tubule? Again, we're supposed to identify the stages of sperm cell development. Diba? So yun ulit, review lang ha. So let's turn to high power. Yeah. Okay. Nice. So this is of course the basal lamina. And as we've said before, that the cell nearest the basal lamina is what we call your spermatogonium. We've already said that there are two kinds of spermatogonium. You have your A and B, which are difficult to distinguish under the light microscope. Again, we'll see the next stage, which is the biggest cell. You can easily see here your genetic material scattered, diba? Within the nucleus. This is now the largest cell. This is now your spermatocyte, specifically the primary spermatocyte. Again, we do not see the secondary form because they rapidly change to the next stage, which is the spermatid. So if you see small round structures very near the lumen, so especially like this, itong maliliit na to, this is now your spermatid. Itong mga maliliit na yan, these are all spermatids. These are all spermatids. If, on the other hand, let me just look for a better one. We now see a grain-like structure like this. No? This will now be your spermatozoa. Okay? So, zoa po, yung pong manipis, small structure. Grain-like structure, that will be your spermatozoa. Now, the other cell we'd like you to identify is a cell that arises from the basement, no? Going to the lumen. It's irregularly shaped. The clue sometimes is your nucleus. That sometimes difficult to see. No? Hanap tayo. Ito. Yan. Can you see that? The shape of the nucleus is elongated, diba? Usually, your spermatogonium has a round nucleus as comparable to this cells over here. No? So ito po, pahaba ang nucleus. So this is an example of your Sertoli cell. Now, what is the Sertoli cell? So usually it's supposed to be from basal lamina to your lumen, okay? And it's the supporting cell because it gives nutrition to your sperm cells. You will find plenty sperm cells surrounding it because the cells are deriving nutrition from this cell. Okay? Ito pa. Tama, galing ni sir, oh. He was able to see this. Very nice. So that's another one. Look at that. Okay? So those are your Sertoli cells. It's really not that difficult. Just look at the nucleus. That is your clue that you are looking at a Sertoli cell. Okay, those are the things I'd like you to identify, most especially the Sertoli, because we have not identified that before. Now the next slide is slide 68. Bakit 68? Sorry. 67, sorry. 67 is your epididymis. Epididymis muna tayo. Now don't be surprised, sometimes you will see epididymis and you'll see on the part somewhere still a part of your testis. Baka makakita kayo ng seminiferous tubules in the same slide. Okay? Baka makakita kayo sa slide nyo. Anyway, this is your epididymis. Again, you will see tubules within. In contrast to the tubules of your testis, notice that the lining is not made up of layers of cells. Diba? Yun ang first clue. You look at the layering of the lining epithelium. Sa seminiferous tubules, we have your so-called seminiferous epithelium. Dito, we have instead a pseudo-stratified columnar epithelium with stereocilia. Let's look at this under low power first. Ayan po. Of course, this is your basement membrane. There you go. This is your pseudo-stratified columnar with stereocilia. Now, this is the only organ with stereocilia in the vas deferens. The vas deferens, though, has only some stereocilia. Ito puro with stereocilia. Okay? Another clue that this is epididymis, there you go, look at the lining, is that within the lumen of the tubule are sperm cells. Bakit po may spermatozoa dito, ma'am? Because the epididymis will serve as a temporary storage for your sperm cells until they reach maturation. So in other words, sperm cells will actually reach maturation in the epididymis. Okay? So yan po. You will see tubules lined by your pseudostratified columnar with stereocilia. And in the lecture, you'll find out that there are actually three kinds of cells here in your lining epithelium. The principal cell, which of course is your columnar cell. Then you're supposed to find your basal cells. Medyo difficult to see yan, basal cell. Basal cells. And of course, the so-called halo cell. Halo cell, if you remember, are what? Bakit halo siya? Why is it called halo? Is it light? Pale? Pale ba yun? It's supposed to be a pale cell with, ano ba siya? Diba siya yung mga lymphocytes? Aralin ulit. Tanong ulit kay Dr. Bardenosa. Okay? So that's the third cell, your halo cell. Now, it's difficult to actually identify your halo cell in the light microscope. Okay? And then, of course, again, another epididymal tubule. Alright? So this is slide 67. So 66 is testis, 67 is the epididymis. The next slide is, of course, the vas deferens, which is the 68. Yeah, 68. Now, the clue that you are in the vas deferens is that you are looking at a single structure with a very thick muscular layer. Ayan, look at that. Nalala niyo yung